Good afternoon, students. In last class, we discussed body fluids and circulation. So, in which how the blood and lymph, which transport nutrients, gaseous constituents in entire body by the help of heart, blood vessels, along with the blood. As also, we discussed certain disorders of blood circulation. So today, in the new synopsis of chapter number 19, excretory products and their elimination. So here, what is excretion? Why we need excretion? So excretion is also a metabolic activity of our body. It is also a metabolic activity of all the living organisms, in which excretion is nothing but a removal of waste nitrogenous products. Waste nitrogenous materials from the body. Why we remove these nitrogenous waste products? How those waste products which are generated in the body? So every day we require energy, the energy required for our metabolic activities. Even excretion is a metabolic activity, it also consumes certain amount of energy. That energy gained by certain nutrients, certain food materials, those food materials are digested. The digested food materials once again burned in the cells to release the energy, during which what we have taken, the food materials, a total amount of food is not completely consumed, in which certain amount of waste products which are eliminated from our body. So like, for example, sodium, that is ammonia, urea, uric acid, and certain minerals, that is mineral ions like hydrogen, potassium, magnesium, like different mineral ions, which are all generated in our body. Those are generated in our body by either digestion of those food. After digestion, what the digested products which are consumed in the body, certain undigested, or certain the minerals which are not required, which are eliminated. So those eliminate either partially, either completely, or completely removal of these materials from our body. So what those removal of such waste materials, waste nitrogenous materials from our body, such phenomenon what we are called as, that is excretion. As excretion of different materials, that is ammonia, either urea or uric acid. So based on The waste nitrogenous products which are converted into either ammonia, either urea or uric acid. Based on these, we grouped different organisms, that is a number of organisms which are grouped into, that is ammonotelic organisms, that is ammonotelic animals, which is nothing but, so what we categorized the name, that is ammonotelic animals, which is nothing but the animals which excrete their waste nitrogenous substance, the waste nitrogenous substance in the form of, that is ammonotelic ammonia. So in the form of ammonia, that is the nitrogen, it is converted into ammonia. This ammonia is excrete. Are you understand? That is ammonotelic animals are nothing but the animals which excrete waste nitrogenous substances in the form of ammonia. In the form of ammonia. See, one thing you keep it in your mind, the ammonia is, it is more toxic material. It is more toxic material. But these more toxic material which require more water to eliminate the ammonia. To remove ammonia from our body, which require more water, because which is the ammonia is readily soluble nature, which is readily soluble nature. These ammonia which remain in the body, which converted into toxic, as hence that ammonia is eliminated from the body. For the elimination of ammonia, elimination of nitrogen in the form of ammonia, it requires more water. So such animals what we are called as, that is ammonotelic animals, especially freshwater fishes. In freshwater fishes, those fish eliminate the waste nitrogenous substance in the form of ammonia, because they live in aquatic condition, they get more water. So as they adapted, that is the ammonotelic animals which require more water. That ammonia is also readily soluble, easily soluble in the aquatic, that is in the water. So such animals what were called as ammonotelic animals. So another group of animals, don't forget, the animals categorized based on the waste nitrogenous substance is converted into different form. Based on that form, we call it as the name of animals, that is the animals which excrete waste nitrogenous material in the form of ammonia, ammonotelic animals. Another one is ureotelic animals. Ureotelic animals. Here, most of the mammals which excrete these waste nitrogenous substances in the form of urea. So urea it is also toxic while it is remain in the body. This urea, it requires less amount of water than this excretion of ammonia. ಅಮೋನಿಯಾ ಫಾರ್ಮ್ ಅಲ್ಲಿ ಎಕ್ಸ್ಕ್ರೀಟ್ ಆಗಬೇಕು ಅಂದ್ರೆ ಜಾಸ್ತಿ ವಾಟರ್ ಬೇಕು ಅಂತ ಹೇಳಿ ಇಟ್ ರಿಕ್ವೈರ್ ಮೋರ್ ವಾಟರ್. ಸೊ ಅದಕ್ಕಿಂತ ಕಡಿಮೆ ವಾಟರ್ ಆದ್ರೂ ಸಾಕು, ದ ಯೂರಿಯಾ ಈಸ್ ರಿಲೀಸ್ಡ್. ಸೊ ಆಸ್ in human, the urea, it is released either in the form of urine, either in the form of sweat. So in both cases, that is sweat and urine, which eliminate the minimum amount of urea, which consists the urine and sweat consist minimum amount of urine. So as such animals, what we are called as ureotelic animals. So another one is uricotelic animals. Uricotelic animals. So uricotelic animals, it is nothing but the organisms or the animals which excrete their waste nitrogenous metabolic products, which convert into uric acid. So means here the nitrogenous substance which convert into uric acid, the uric acid is eliminated. So these uric acid elimination which require minimum water. So very less amount of water is required to elimination of uric acid through the excretory system of such animals. So example, that is terrestrial amphibians, reptiles, even aves, insects. So which are all those which eliminate these organisms eliminate their waste nitrogenous materials in the form of uric acid. As the animals which are categorized by these different groups based on what they are eliminate the waste nitrogenous substance, nitrogen content, which convert into either ammonia, either urea or uric acid. So for elimination of these contents or waste nitrogenous substances which require must and should there is an excretory system or the excretory organs, that is excretory organs. So these excretory organs, as you know, in the simple, that is a lower organisms which does not contain any modified or well organized excretory organs. As in certain invertebrates, which consist simple tubular forms. Simple tubular organs, invertebrates, for example, annelids, enriched earthworm, which consist excretory organ called nephridia. Before that, that is platyhelminthes, in which that is planaria, important, which consist plain cells. These plain cells are excretory cells or excretory structures, which are also called as protonephridia. So even that is cephalocardiaca, that is Amphiaxis, it consists protonephridia. Protonephridia, it is nothing but first form of a simple nephridia. So these protonephridia is, that is the excretory organ of that is cephalocardiacs. Phlegm cells are excretory cells of platyhelminthes or planaria. Nephridia are excretory structure of earthworm, excretory organ of earthworm. So even certain insects like cockroach, as we discussed in previous classes, cockroach consists, that is Malpighian tubules. Most of the insects which consist these small hair-like structures which are present in the, that is hindgut and midgut junction. There is presence of nearly more than 100 to 150 Malpighian tubules present in cockroach. The Malpighian tubules are excretory organs in insects. Those insects, in that insects, the Malpighian tubules which absorb the waste nitrogenous product and which converted into uric acid, that uric acid released into hindgut. So like the different types of the excretory organs which are present in the, like these invertebrates. But in vertebrates, which are complex tubular forms or complex tubular organs. Complex tubular organs. So these complex tubular organs, which are nothing but most of the kidneys. Kidney is the most a complex tubular structure. Those are excretory organs in most of the vertebrates. So as we discussed that is human excretory system. Human excretory system. In human excretory system, which consists a pair of kidneys, a pair of ureter, blood vessels, urinary bladder. Urinary bladder. So here, as you seen in the diagram, a pair of kidneys. Here, little bit higher position in the left kidney, a small height, which are located in abdominal cavity, just below the abdominal cavity, as the kidneys which are present in the lateral portions, which is having a pair of blood vessels, that is a pair it is nothing but one of the tube, it is inferior vena cava, means these inferior vena cava which collect the deoxygenated blood from the kidneys and which supply to the heart. As another blood vessel, that is dorsal aorta, which brought the oxygenated blood from the heart which supply to the kidneys, and a pair of ureter which originated from the kidneys, that is a notch-like structure of kidney, it is called as hilum. From the hilum there is origination of these a pair of ureters, each ureter arising from a kidney, which collect the urine, what it produces the urine by kidneys. Those ureters which link with urinary bladder. The urinary bladder, storage function of urine, what passing the urine from, passes of urine from kidneys to urinary bladder through the ureter, it collected. After filling up this urinary bladder, there is elimination of urine by a pore called urethra. That urethra is guarded by a muscles called urethral sphincter muscles, which relax and release the urine. As it consists, the human excretory system, it consists a pair of kidneys, a pair of ureter, blood vessels, and urinary bladder. So while we discussed in these each, in human, a sagittal section of kidney. So here, the kidney, which is nearly 10 to 12 centimeter length, 10 to 12 centimeter length, 5 to 7 centimeter width, 2 to 3 centimeter in thickness. So each kidney is a reddish brown colored structure. A reddish brown colored organ, which is bean shaped structure. Bean shaped structure. So it weighs nearly 120 grams to 170 grams, that is weight of kidney. As the kidney, which covered with a tough layer, that is double membrane structure. The entire kidney, it is divided into two parts. The portion of this entire kidney, it is divided into two parts, that is outer medullary region, outer cortex region, inner medullary region, that is outer cortex region and inner medullary region or inner medulla. Outer cortex, what the portion of these? So this from here to here, it is totally medullary region, inner part. So in the cortex region, there is presence of Bowman's capsule of nephron. Like each kidney, it consists near 1 million nephrons. 1 million nephrons. That is, nephron is structural and functional unit of kidney. Nephron is structural and functional unit of kidney. So as a notch-like structure, ಸಣ್ಣ ಗುಡಿ ತರ ಇರುತ್ತೆ. So as it has two faces. It is convex face, it is concavity face. The concavity face, which consists a notch-like structure, it is called hilum. So the parts which is the parts of kidney, it is also starts from renal. That is hilum. It is a notch-like structure of kidney where the origination of ureter and blood vessels. So ಇಲ್ಲಿ diagram clear ಆಗಿ ನೋಡಬಹುದು. It is the origination of ureter and even the origination of both renal vein and renal artery. Renal vein which supply the oxygenated, renal artery which carry the deoxygenated blood. So both blood vessels which originate from the notch and the ureter is also originated from the hilum position. It is what we are called as that is hilum. So those ureter origination from the hilum and it followed by a large space. This entire vacant space, it is called as renal pelvis. This space, it is renal pelvis. The renal pelvis, which little bit elaborate into the medulla, what the portion it is called as these portions which are called as renal calyx. So the portions like these which are called as renal calyx or renal calyces. Renal calyces or renal calyx. On the renal calyx there is presence of a pyramid shaped structure. Pyramid shaped structures. So these are the pyramid shaped structures which are called as renal pyramid. Renal pyramid or medullary pyramid. Renal pyramid or medullary pyramid. In that renal pyramid or medullary pyramid, which consists a number of nephrons. But most of the nephrons which are present in the renal pyramids, in which the Bowman's capsule of nephron which are present in the cortex region, but remaining part of renal tubules which are totally present in the renal medullary region. So here we divide the two types of that is the kidney, that is nothing but the two types of nephrons there is presence. Two types of nephrons. Here, in which kidney? In the kidney, if the renal tubule which are short or length. So these renal tubules of nephron which are long, which are called as juxtamedullary nephrons. Juxtamedullary nephrons, which consist long renal tubules. So those which present in the deep position of renal medulla. Certain nephrons which has short length of these renal tubules, just which are inserted in the medullary region. ಈಗ ನೋಡಬಹುದು ಈ ಡಯಾಗ್ರಾಮ್ ಅಲ್ಲಿ ಈ ಟ್ಯೂಬ್ಯೂಲ್ಸ್ ಜಸ್ಟ್ ಇನ್ಸರ್ಟ್ ಆಗಿದೆ ಮೆಡುಲರಿ ರೀಜನ್. ಇಲ್ಲಿರ್ತಕ್ಕಂಥ ನೆಫ್ರಾನ್ ಟ್ಯೂಬ್ಯೂಲ್ಸ್ ಜಾಸ್ತಿ ಒಳಗಡೆಗೆ ಎಂಟರ್ ಆಗಿರ್ತಕ್ಕಂಥದ್ದು. ಸೊ ಇವನ್ನ ಜಕ್ಸ್ಟಾ ಮೆಡುಲರಿ ನೆಫ್ರಾನ್ಸ್ ಅಂತ ಹೇಳಿ ಕರಿತೀವಿ. ಸೊ ಈ ತರ ಎ ಶಾರ್ಟ್ renal tubules of nephron, such nephrons are called as cortical nephrons. Cortical or corticoid nephrons, which consist short. So as a thick layer is surrounding the kidney, which is called as renal capsule, a thick layer or double layer. So it is the entire structure of human kidney. A kidney is reddish brown structure which covered with the renal capsule, a tough layer. It has two faces, concavity face and convex face. In the concavity face there is origination of ureter and blood vessels, that is hilum, which followed by inner there is vacant space in the hilum region, there is renal pelvis. From the renal pelvis there is origination of ureter. The renal pelvis which elaborate a structures called, that is renal calyx. In between renal calyx, so each other, in between renal pyramids, there is space. Such space it is called as renal column or columns of Bartini. The space present in between the two renal pyramids, which is called as renal column or columns of Bartini. As the entire kidney, outer cortex, inner medulla, most of the nephron part which is present in the medullary region, only the Bowman's capsule of nephron present in the renal cortex region. So as most of the nephrons which are present in the renal pyramids, so it is just the structure of kidney. So each kidney which consists a number of structural and functional units called nephron. Nephron is structural and functional units of kidney. While we recording, we know the entire structure of nephron. The nephron, it consists different parts, that is Bowman's capsule, glomerulus, afferent, and efferent arteriole, afferent and efferent arteriole, PCT, that is proximal convoluted tubules, Henle's loop, DCT, that is distal convoluted tubule, and collecting duct. So even the blood vessels which runs parallel to these renal tubules, which are called as vasa recta. So like the first one, the Bowman's capsule. The Bowman's capsule is a cup-shaped structure. It is the Bowman's capsule, a cup-shaped structure. It is called as a Bowman's capsule. Second one is a glomerulus, a number of blood vessels which are combined together to form a knot-like structure. That is what they are called as glomerulus. The entire glomerulus which are present inside the Bowman's capsule, what the cup-shaped structure of the nephron, that is Bowman's capsule. So those glomerulus which is arising from an afferent arteriole. So this afferent arteriole is which is larger. The size of the tube is little bit large than that is efferent arteriole. The blood entered into glomerulus through a large afferent arteriole, and which eliminate or with that blood exit from the glomerulus is a narrow. So means a wider tube, that is afferent arteriole, a narrow tube, it is efferent arteriole. And the Bowman's capsule, which is followed by a tube called a coiled tube. That coiled tube, it is called as, that is proximal convoluted tubule. Proximal convoluted tubule. And this proximal convoluted tubule, which consists, that is hairpin curve. So this hairpin curve, it is nothing but it consists descending limb, that is from the proximal convoluted tubule, a limb which is downward, that is descending limb, and a loop formed, that is hairpin curve. It is the hairpin curve, Henle's loop, and ascending limb. So the ascending limb, the same hairpin curve which is ascends. These ascending limb which connect the distal convoluted tubule. So it is also a thick, a coiled structure. It is the distal convoluted tubule. The distal convoluted tubule which connect to a connecting duct. Each connecting duct which connected with a number of nephrons. The connecting duct which collect the urine from a number of nephrons. Because nearly 1 billion nephrons present in the kidney. Most of those collecting ducts which connected with a various number of nephrons. So, vasa recta, it is also an important part present in the structure of nephron. Students, see here there is a light colored. So, there is which runs parallel to the blood vessel. Parallel to the tubules of nephron. PCT, that is proximal convoluted tubule, DCT, distal convoluted tubule, and the hairpin curve, that is Henle's lobe. ಈ ಮೂರು ಟಿಬ್ಯೂಲ್ಸ್ ನ ಪಕ್ಕದಲ್ಲಿ ನಂಬರ್ ಆಫ್ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಪಾಸ್ ಆಗಿರುತ್ತೆ ಪ್ಯಾರಲಲ್ ಆಗಿ. ದಟ್ ಪ್ಯಾರಲಲಿ ಪಾಸಿಂಗ್ ಆಫ್ ದೀಸ್ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ವಿಚ್ is called as vasa recta. It is also an important, it leads to formation of a concentration of urine takes place in kidney. As the vasa recta, which the blood vessels which runs parallel to the nephron. So in the nephron, the Bowman's capsule where the blood is filtrated. So we discussed that is what is the how the urine is formed. How the urine is formed. So next, the urine formation. So just before that, just we discussed what is the functions of the tubules of these. Next we go to that is urine formation. So here the first one, proximal tubules. Proximal convoluted tubules. These proximal convoluted tubules reabsorb the reabsorption of nutrients. Reabsorption of nutrients, that is nearly 70 to 80% of nutrients and electrolytes, and even secretion of hydrogen ions and phosphate ions and certain amount of ammonia in filtrate. Filtrate ಗೆ secrete ಮಾಡೋದು. Filtrate secrete ಮಾಡೋದ್ರಿಂದ it helps to ionic balance maintain ಮಾಡುತ್ತೆ and to maintain pH also. So reabsorption of nutrients and electrolytes by the proximal convoluted tubule, secretion of certain ions to maintain ionic balance and to maintain pH of interstitial fluid, the fluid present in the kidney. As Henle's lobe, the Henle's lobe which the reabsorption is very minimum. A very less amount of reabsorption occurred in the Henle's lobe. Here it maintained osmolarity. It maintained osmolarity of interstitial fluid present in the kidney, and DCT, that is distal convoluted tubule, so these distal convoluted tubule reabsorption of sodium and water. Reabsorption of sodium and water and secretion of hydrogen ions and hydrogen ions, and that is potassium ions. So ions secreted into filtrate. Filtrate secrete ಮಾಡ್ತಕ್ಕಂಥದ್ದು. And collecting duct, which is also reabsorption of water. Reabsorption of water. So most of the water which reabsorbed by the collecting duct, while the filtrate passes through the collecting duct. So because if maybe the body fluids are lost more, then it reabsorbs more water. Body fluid concentration, body fluid ಅಲ್ಲಿ water concentration ಕಡಿಮೆ ಆಯಿತು ಅಂದ್ರೆ ಇದು reabsorb ಮಾಡ್ಕೊಳ್ಳೋದು. ಜಾಸ್ತಿ ಇತ್ತು ಅಂತಂದ್ರೆ secrete ಮಾಡ್ಬೋದು. So even the urea, that is urine consists more urea, it is also required to osmotic balance as even which secreted that urea into the, that is interstitial fluid present in the kidney. So like there is different functions of these tubules, that is PCT, headless lobe and distal convoluted tubules. So next, formation of urine. So how the urine form? In the kidneys, there is concentrated urine is formed. These concentrated urine formed by the help of nephrons, that is 1 billion nephrons present in the kidney, in which the urine formation takes place under 3 steps. The first one, it is filtration of blood, that is what we are called as ultrafiltration. Filtration of blood, it is also called as glomerular filtration. Glomerular filtration. Second one is tubular reabsorption. Tubular reabsorption. Third one, tubular secretion. First one, glomerular filtration or filtration of blood in the first step. Second step, tubular reabsorption. Third step, tubular secretion. So here, the filtration of blood, in which glomerular filtration, the glomerular filtration is takes place in the glomeruli of Bowman's capsule. The glomeruli present in the, that is glomeruli, it is nothing but a number of blood vessels which combine together to form a knot-like structure. That is what we are called as glomerulus. In the glomerulus, the blood entered through a wider afferent arteriole, means there is a large amount of blood entered into the glomerulus. As what the blood entered into the glomerulus or in the Bowman's capsule, the blood is filtered. The blood filtered. The filtrate passes into the Bowman's capsule and renal tubules. The remaining amount of blood which exit from these glomerulus by a narrow afferent arteriole. So just a blood entered through wider tube, the blood exit from a narrow tube. So in these glomerulus, in the glomerulus, there is creation of more pressure. The pressure is created because a wider tube carry the blood, a narrow tube carry the blood from the glomerulus. It causes more pressure, that is glomerular pressure. Glomerular pressure. By presence of more glomerular pressure, it helps to filtration of the blood. Filtration of blood. At the time of filtration, all the contents of plasma, except certain proteins, So because these proteins are little bit larger sized particles which are not entered through the slit pores present in the Bowman's capsule. So hence such filtration is also called as ultrafiltration. Ultrafiltration. This filtration, which is carried by 3 layers. Which are the 3 layers means? See here, it is the endothelium of glomerulus, that is the blood vessel, and epithelium of Bowman's capsule. Epithelium of Bowman's capsule. For example, it is the blood vessel, it is the epithelium of Bowman's capsule. Sorry, endothelium of Bowman's capsule, epithelium of that is Bowman's capsule, endothelium of glomerulus. In between these presence of a space, it is called as little bit basement membrane. So these basement membrane which helps to attachment of endothelium of glomerulus or blood vessel, that is epithelium of these Bowman's capsule. Through this, all these 3 layers, there is filtration is occurred. But in the Bowman's capsule, the epithelial cells, most of those epithelial cells which are called as the podocytes, these podocytes, it is what we are called as podocyte, that is the epithelial cells of Bowman's capsule. In between these epithelial cells, there is a space present. This space, what we are called as slit pore. Through these slit pores, only a minute substance will pass, but larger proteins are not passed. Hence, the proteins of plasma is not passed here, but most of the nutrients will enter. So that is what the filtrate which are passes into the Bowman's capsule, that entire phenomenon what we are called as glomerular filtration. So these glomerular filtration, it is nearly 125 ml of filtrate formed by kidneys. That is 125 ml per 1 minute. So what is the amount of blood is filtrated here? So nearly 1100 ml to 1200 ml of blood filtered by kidneys per 1 minute. So, what is the amount of filtrate formed per minute by both kidneys? It is called as glomerular filtration rate. Filtration rate, that is nothing but GFR. GFR is nothing but glomerular filtration rate, that is 125 ml per minute. Here 1100 to 1200 ml of blood filtered by kidneys per 1 minute. So yesterday we discussed heart, in which one ventricle pumped 5040 ml of blood. So istu blood anna pump ಮಾಡುತ್ತೆ ಅಂತಂದ್ರೆ ಯಾವಾಗ blood per 1 minute. 1 ನಿಮಿಷಕ್ಕೆ 5 liters of amount of blood will be pumped by one ventricle. So ಇದರಲ್ಲಿ ಈ amount ತಗೊಂಡಾಗ only 1/5 part of blood which will be filtered by kidneys per minute, in which there is formation of 125 ml of filtrate formed by both kidneys. It is nothing but in one day there is 180 liters of filtrate formed per day. On dinike 180 liters of glomerular filtration formed. The filtrate entered into the Bowman's capsule. So it is the first step of glomerular filtration. Glomerular filtration is also called as ultrafiltration because the slit pores which not enters the larger molecules, that is the proteins of plasma. The remaining all plasma contents which will be filtered. So the second one is tubular reabsorption. Tubular reabsorption. So ಇಲ್ಲಿ glomerular filtration ಅಲ್ಲಿ 180 liters of filtrate formed per day ಅಂತಂದ್ರೆ 180 liters per day. So 180 liters filtrate ಆಗ್ತಾ ಇದೆ ಅಂತಂದ್ರೆ ಎಲ್ಲಾನು ಯೂರಿನ್ ಕನ್ವರ್ಟ್ ಆಗತ್ತಾ? ಇಲ್ಲ. So just we pass out nearly 1 to 1.5 liter per day. That is the urine eliminated by a person, that is 1 liter to 1.5 liter. 1 to 1.5 liter. That is nothing but nearly 90% of filtrate reabsorbed by renal tubules of nephron. So nephronally, the renal tubules are nothing but proximal convoluted tubule, endless loop. It consists ascending limb, hairpin curve, ascending limb, descending limb, hairpin curve, ascending limb, and distal convoluted tubule, and even the collecting duct. So it's all involved in the reabsorption. That is reabsorption of 99% of filtrate entered into the Bowman's capsule. That is nothing but the filtrate formed in the Bowman's capsule entered into proximal tubule, and it passes through the proximal tubule, proximal convoluted tubule, endless loop, and that is distal convoluted tubule, during which most of the contents of these filtrate which entered once again into the blood vessels. Blood vessels ಗೆ enter ಅಂದ್ರೆ reabsorption ಆಗುತ್ತೆ. 99% of filtrate is reabsorbed by these entered into the blood vessels. That is vasa recta. The next, that is a tubular secretion. That is tubular secretion in which only hydrogen ions, potassium ions, which filtered even the water content, that is secretion. If the urine contain more concentrate, then there is secretion of water is also occurred. So as the tubular secretion which helps to maintain ionic balance of urine, as well as it maintain pH, it maintain acid-base balance. Acid-base balance, ionic balance, a pH concentration, a pH of, that is hydrogen concentration, it is the ionic concentration. So because hydrogen ions which are secreted by the distal convoluted tubule and collecting duct as it required. So at the end there is only concentrated urine, it is passes into the collecting duct, and these collecting duct release the urine into the renal pelvis. Renal pelvis. So as like the urine is formed in both kidneys, nearly 1.5 liter of urine passes per day. That urine is very, very concentrated, while we compare to, that is the filtrate of Bowman's capsule. So next, mechanism of concentration of the filtrate. So how the filtrate which is more concentrated, which is occurred. See, the diagram shows what type of reabsorption is occurred in the renal tubules, what type of molecules which are secreted by the renal tubules, what type of molecules which eliminate from the descending limb and ascending limb. As in this diagram, it is also shows, that is how the filtrate formed, how the concentrated urine formed in the nephron. While in the filtrate passes to the proximal convoluted tubule, the reabsorption of sodium chloride, water, that is salts, water, nutrients, which are all entered into the blood. If maybe certain amount of hydrogen ions and ammonia is secreted into the filtrate at the portion of proximal convoluted tubule. From here to here it is the Henle slope. The Henle slope consists of descending limb, hairpin curve, and ascending limb. The membrane of descending limb is permeable to water. It is nothing but water content, it is move out from the filtrate, but it does not impermeable to sodium chloride. impermeable, ಇತರ ಬರ್ಲಿಕ್ಕೆ ಚಾನ್ಸ್ ಇಲ್ಲ. But the membrane of ascending limb, it is permeable to sodium chloride, that is salts, but water is it is not move out from the upper membrane, that is ascending limb membrane. It is impermeable to water. So based on these concentration, or it is also called a little bit counter current flow, as in the distal convoluted tubule, it is also reabsorbed by sodium chloride, that is salts or water and bicarbonates, and it secretes potassium and hydrogen ions into the filtrate. So little amount of urea is entered into the ascending limb. If the urea content more present in the urine, it is once again released from the collecting duct. Even water concentration is more in urine, that is urine, in which water is reabsorbed in the portion of collecting duct. So here the diagram which shows that is how the mechanism of concentration of filtrate is formed. It is the vasa recta, it is the portion of nephron. So in the kidney, there is a portion, it is outer cortex, it is inner medulla. Outer cortex region, it is inner medullary region. See here, it is the cortex region, it is totally medullary region. In the cortex, outer cortex, it is inner medullary region. In the inner medulla, outer medulla, इतर ಬೇಕಾದರೂ ನಾವು ಡಿವೈಡ್ ಮಾಡ್ಕೋಬಹುದು. It is the outer cortex. At the time of in the cortex region, already we discussed there is presence of only Bowman's capsule. The most of the Bowman's capsules which are present in the cortex region, the remaining renal tubules which are totally present inside the medullary region. So in which there is presence of interstitial fluid or medullary fluid present in the entire kidney. So in the fluid present in the cortex region, there is 300 molar osmolarity. It is depend upon presence of mineral concentration in that interstitial fluid of kidney. But inner to medulla, there is 1200 molar osmolarity per liter. Osmolarity per liter. Osmolarity liter. That is more mineral concentration present here less. That is 300 moles osmolarity of per liter. So ಇಲ್ಲಿ ಕಡಿಮೆ ಹೋಗ್ತಾ ಜಾಸ್ತಿ ಆಗಿದೆ. So it is helps, what the diagram shows, it helps to how a concentration of filtrate formed in the kidney or concentration of filtrate formed in the renal tubules of nephron. just you know that the filtration formed in the kidney, it is depend upon association of vasa recta and nephron. That is to maintain the concentration of filtrate formed in the kidneys by both vasa recta and nephron. It is called as counter current mechanism. So counter current mechanism, it is nothing but in the nephron, the fluid which move this direction parallel to vasa recta, the fluid which runs upward direction, that is opposite direction. The filtrate direction in the nephron, the reabsorption content moved in the blood vessels, it is opposite direction. Hence, the opposite direction flow of the filtrated blood which is present near the limbs of nephron and the limbs of blood vessels, that is vasa recta, that mechanism what we are called as counter current mechanism. So here the counter current mechanism, there is important sodium chloride and urea which running opposite direction in the filtrate and that is in the blood. For example, in the descending limb, the membrane of descending limb which is permeable to water, impermeable to sodium chloride. But certain amount of water sodium chloride which entered into the descending limb of, that is vasa recta. So at the time of ascending limb, what it released, the sodium chloride which is once again released into in the ascending limb of vasa recta, it is once again released. Descending limb alli sodium chloride ತಗೊಳ್ತಾ ಇದೆ, ascending limb alli ಬಿಟ್ಕೊಡ್ತಾ ಇದೆ, ಅಂದ್ರೆ ರಿಲೀಸ್ ಮಾಡ್ತಾ ಇದೆ, ಸೆಕ್ರೇಟ್ ಮಾಡ್ತಾ ಇದೆ, ಆಚೆಗೆ ಬಿಡ್ತಾ ಇದೆ. So as like the sodium chloride maintain, that is osmolarity is maintained in between, that is ಇಲ್ಲಿ ನೋಡಬಹುದು ರಿಲೀಸ್ ಆಗ್ತಾ ಇದೆ, ಇಲ್ಲಿ ನೋಡಬಹುದು ಸೇಮ್ ಸೋಡಿಯಂ ಕ್ಲೋರೈಡ್ ಎಂಟರ್ ಆಗ್ತಾ ಇದೆ, ಅದೇ ಸೋಡಿಯಂ ಕ್ಲೋರೈಡ್ ಮತ್ತೆ ರಿಲೀಸ್ ಆಗ್ತಾ ಇದೆ. So it is depend upon to maintain osmolarity ಇಲ್ಲಿ ಮತ್ತೆ ರಿಲೀಸ್ ಆಗಿದೆ. So like while it is in the deep, that is in the cortex to medullary region, in the medullary region there is 1200 molar osmolarity per liter, that is increase the osmolarity in the medullary region, that is interstitial fluid is what it formed, it is moved the fluid, that is filtrate formed towards the collecting duct is little bit more concentrate. And even the urea is also, that is exchanging between nephron and vasa recta, that is depend upon, that is certain amount of urea is entered into ascending limb of ಇನ್ನೊಂದು diagram ಅಲ್ಲಿ ನೋಡಬಹುದು ನಾವು. See here the same urea content which entered into the ascending limb. If urine consists more urea, the same urea content it is secrete, that is reabsorbed into the blood vessels. As like the counter current mechanism, it helps to formation of a concentration filtrate. There is formation of concentration urine formed in the both kidneys. Next, Regulation of kidney function. So kidney which helps to formation of urine. Sometimes formation of urine is less at the time of summer, formation of urine is more at the time of winter, it is little bit variate. Those urine formation is variate depend upon what the filtrate formed in the Bowman's capsule. Like the kidney function is regulated by the different organs, that is hypothalamus of brain, juxtaglomerular apparatus which is present in the kidney, and even heart is also regulate the kidney function. So here hypothalamus, it is present in the midbrain. Sometimes the glomerular filtration is decreased. The glomerular filtration is decreased. Then activate osmoreceptors. Activate the osmoreceptors. Osmoreceptors are nothing but which receive the osmolarity of body fluids. As the osmoreceptors, that is nothing but the glomerular filtration is decreased. That is, glomerular filtration is nothing but glomerular filtration rate is less. ಕಡಿಮೆ ಆಯಿತು ಅಂತಂದ್ರೆ ಯೂರಿನ್ ಫಾರ್ಮೇಷನ್ ಕಡಿಮೆ ಆಗ್ಬೋದು. But commonly it's working not normal. So then automatically these osmoreceptors are activated. Means they activate and receive the signal. So by that signal, those activate the cells of hypothalamus to secrete ADH hormone, that is anti-diuretic hormone. Anti-diuretic hormone, it acts as a vasopressin. So these hormone which is secreted by neurohypophysis. So when GFR is fall down, because there is not more formation of pressure in glomerulus. ಜಾಸ್ತಿ ಗ್ಲಾಮೆರುಲರ್ ಪ್ರೆಷರ್ ಜಾಸ್ತಿ ಆಗಿರಲ್ಲ. ಯಾವಾಗ ಗ್ಲಾಮೆರುಲರ್ ಪ್ರೆಷರ್ ಕಡಿಮೆ ಆಗುತ್ತೋ ಗ್ಲಾಮೆರುಲರ್ ಫಿಲ್ಟ್ರೇಷನ್ ಕೂಡ ಕಡಿಮೆ ಆಗುತ್ತೆ. So automatically the hypothalamus secrete the antidiuretic hormone. It acts as a vasopressin. It is nothing but vasopressin is nothing but which constrict the blood vessels of glomerulus. That is, vasopressin constrict the blood vessels of glomerulus. ಯಾವಾಗ ಗ್ಲಾಮೆರುಲಸ್ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಕನ್ಸ್ಟ್ರಿಕ್ಟ್ ಆಗುತ್ತೋ ಅವಾಗ ಆಟೋಮ್ಯಾಟಿಕಲಿ ಪ್ರೆಷರ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ ಒಳಗಡೆ. ಗ್ಲಾಮೆರುಲಸ್ ಅಲ್ಲಿ ಪ್ರೆಷರ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ. ಯಾವಾಗ ಪ್ರೆಷರ್ ಜಾಸ್ತಿ ಆಯಿತು ಅವಾಗ ಕಾಮನ್ ಆಗಿ ದ ಫಿಲ್ಟ್ರೇಟ್ ಈಸ್ ಫಾರ್ಮ್ಡ್ ಮೋರ್ ಇನ್ ದ ಬೌವಲ್ಸ್ ಕ್ಯಾಪ್ಸೂಲ್. So these neurohypophyses which secrete the antidiuretic hormone as in glomerulus in which pressure is constrict the vessels of glomerulus, automatically increase the GFR. So glomerular filtrate rate is automatically increased. ಯಾವಾಗ increase ಆಗುತ್ತೋ so filtrate form ಆಗ್ತಕ್ಕಂಥದ್ದು ಕೂಡ increase ಆಗುತ್ತೆ. ಯಾವಾಗ filtrate increase ಆಯಿತು it is normal urine formed in the kidneys, as like the thalamus which regulate the kidney function. So another second one, that is JGA apparatus, that is juxtaglomerular apparatus. Juxtaglomerular apparatus. It is nothing but a sensitive area where afferent arteriole and distal convoluted tubules connect or attach. Afferent arteriole and distal convoluted tubule, where it is the contact region, which is called as juxtaglomerular apparatus. So means ಅಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ cells, the cells of juxtaglomerular area, suddenly these cells which release renin hormone, which released renin hormone. So ಯಾವಾಗ renin hormone secrete ಆಗುತ್ತೆ ಅಂದ್ರೆ, if GFR is fall down, ಮತ್ತೆ ಸೇಮ್ ಆಯಿತು. ಹೇಗೆ GFR ಕಡಿಮೆ ಆಯಿತು, ಹೈಪೋಥಾಲಮಸ್ ಸೆಲ್ಸ್ ಆಕ್ಟಿವೇಟ್ ಆಗಿ ಡೈಯೂರೆಟಿಕ್ ಹಾರ್ಮೋನ್ ಸೆಕ್ರೀಟ್ ಆಗಿ ಗ್ಲಾಮೆರುಲಾರ್ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಅನ್ನು ಕನ್ಸ್ಟ್ರಿಕ್ಟ್ ಮಾಡಿ ಇನ್ಕ್ರೀಸ್ ದ ಜಿಎಫ್ಆರ್ ಇನ್ಕ್ರೀಸ್ ಮಾಡುತ್ತೆ. ಅದೇ ತರ ಇಲ್ಲೂ ಸಹ ಅಷ್ಟೇ, ಯಾವಾಗ glomerular filtration rate is fall down, automatically these JGA cells are activate which secrete the renin hormone. So ಏನು ಕೆಲಸ ಮಾಡುತ್ತೆ, ಹೇಗೆ ಕೆಲಸ ಮಾಡುತ್ತೆ ಈ renin hormone? So the renin hormone secreted by these juxtaglomerular cells which convert Angiotensinogen. It is an inactivated protein present in the blood. So these angiotensinogen, it converts into angiotensin 1. Once again, these angiotensin 1, it is converted into angiotensin 2. So these angiotensin 2 is a powerful vasoconstrictor. Angiotensin 2 is a powerful vasoconstrictor which constricts the blood vessel. Automatically the blood vessels of glomerulus are constricted. There is normal GFR. That is, glomerular filtration rate is increased. As it increased, there is commonly formation of normal amount of urine is formed. So, as like the JGA apparatus which are working, which regulate the functioning of kidney. So another one is heart. So heart is a double pumping organ as we discussed it yesterday, which helps to regulate the kidney function. So as at the time of more, the GFR concentration is more, then automatically heart is secreted ANF. It is nothing but atrial natriuretic factor. The walls of heart secrete atrial natriuretic factor. This atrial natriuretic factor is a vasodilator. So means if GFR is increased, suddenly heart muscle secrete atrial natriuretic factor. It is a vasodilator. Automatically dilation of blood vessels. So when dilation of blood vessels, automatically decrease the GFR. So when the glomerular filtration rate is decreased, automatically it comes to normal as the GFR is decreased. Decrease the GFR, that is normal formation of urine formed. So as well, the regulation of kidney function by hypothalamus, JGA cells and heart. So remaining part we continue in the next class.